My name is Sean Escola. I'm an assistant professor at Columbia, and I'm one of the on the program committee for Neuromatch. And we're really excited to welcome Genevera Allen from Rice University to speak with us today, this morning. So Genevera did her PhD with Robert Tibshirani at Stanford, and then she took on a faculty position at Rice, where she's in the electrical engineering and computer engineering departments, and she has an affiliation with Baylor College of Medicine also, where she does where she interfaces with some of the neuroscience faculty there, and she's working on applying machine learning approaches to analyzing neuroscience data, among other things, and very excited to get an update from you this morning.
Cool, thanks so much, Sean. Thanks everybody for joining early in the morning here / other time zones at different times. So I'm excited to talk to you today about graph quilting. And this is a new, and you've probably never heard of graph quilting before because it's completely new. And this is a newer project in my group that I'm excited to share with you. So first, this is joint work with my collaborators, Giuseppe Vinci and Gautam Desarthi, both of whom were former postdocs at Rice University and now have faculty positions elsewhere. They're great collaborators on this project, especially Giuseppe, who is now at Notre Dame. So our motivation is to study neuronal connectivity. And you can break this down, there's really two types of connectivity: structural connectivity, anatomical connectivity. This is the physical connections, the wiring structure. But of course, to get that type of data is in vitro. It's very intensive, very expensive to get that type of data. We'd love to kind of have more large-scale anatomical neuronal connectivity data, hard to get. Another type of connectivity that is perhaps easier to get an estimate of is functional neuronal connectivity. And this is studying the statistical dependence between neurons based on their activity traces. These are typically done in vivo in animals while they're performing tasks or exhibiting some type of behavior. And in this talk, we're going to specifically look at functional connectivity via graphical models, which I'll introduce in a little bit later. But graphical models are basically probabilistic distributions modeling dependencies between data. In particular, we're going to take every vertex or every node in our graph to be one of the neurons that we're recording from or imaging. And every edge between neurons will denote a conditional dependence relationship between two neurons. So this is one way of modeling the kind of statistical dependencies that denote functional connectivity data. These are typically estimated from large-scale experiments where animals are doing specific tasks. You might ask, why are we interested scientifically in functional connectivity? Well, first we might say, hey, anatomical connectivity is really hard to get. Can we use functional connectivity as a proxy for anatomical connectivity? This is largely unknown, but is potentially an intriguing possibility. We might want to understand how functional connectivity changes in response to stimuli or to produce specific behaviors in animals. We might also want to understand what are just the larger scale patterns of functional connectivity. Are these revealing certain properties of neural networks? Do those properties change in response to various stimuli and so forth? So there's a lot of scientific questions here, and this is a relatively unexplored type of area, especially for large-scale type recordings. So just to briefly review first before we move on, probabilistic graphical models. These are fantastic and fun machine learning models to work with. They're probability distributions where every relationship is a conditional dependence relationship, and this is denoted by edges in a graph structure. So we can visualize this probability distribution as a graph or as a network. And we're specifically today going to study Markov networks, which are undirected graphical models. In Markov networks, an edge between two nodes denotes a conditional dependence relationship, where a lack of an edge denotes conditional independence. So for example, these are a very strong form of dependency, and these are very kind of rich class of probabilistic models, because if we're looking at mere correlation or associations, say if neuron C is really kind of connected to A and B and is really driving the activity seen in A and B, then you expect C, A, and B to all be correlated. And in fact, if you look at the correlations, you would probably see this network structure. But by conditioning on C, we really see that A and B aren't actually related. They're only related through C. So this allows us to draw much more stronger forms of inference about functional connectivity, and is a really rich class of models to use. So probabilistic graphical models have been widely used in lots of areas. They're used commonly in human neuroimaging data, modeling functional connectivity. I've done some work in this area previously. They're widely used in genomics and systems biology. I've done a lot of work in this area of modeling kind of genomic networks and interactions, and using these to discover potential biomarkers and pathways in genomics. So these are very widely used models in a lot of different fields. They're maybe less widely used to model functional neuronal connectivity, but they're very closely related, and I'm not going to go into all the details, to commonly used methods to describe functional connections, such as partial correlations, Ising models, or GLMs. So these are kind of used, but maybe not the full kind of the full machinery that we have in machine learning for probabilistic graphical models. But there are certainly some challenges when it comes to applying these types of models to large-scale neuronal recording data. And specifically, I'm listing some of the challenges here. My group has been working on various aspects of these. We've got non-Gaussian, we've got very spiky data, we need to somehow account for stimuli and behavior. We, of course, are only recording from a subset of neurons in the brain. There's probably latent effects from other neurons and so forth. It's very large data. There's all sorts of challenges here but I'm not going to address most of these today. I really want to focus on one specific challenge, and that is non-simultaneous recordings. So specifically, and this is in collaboration with Andreas Tolias's lab at Baylor College of Medicine, he's trying to record from 100,000 neurons in a 1 millimeter cubed portion of the brain. So this kind of brain cube that's happening here, and in the visual cortex. And the goal is to image all of these neurons and estimate the functional connectivity between 100,000 neurons in the visual cortex, okay, using 2 photon calcium imaging. So this is our brain cube, but there's a slight challenge with this. There's a trade-off kind of with these technologies between the temporal resolution you can get and the number of neurons that you can record from simultaneously. And so traditionally, because of this, you need enough temporal resolution to actually see spiking and activity patterns that most people actually record in horizontal layers within the brain. So you might have a recording like this. You can't record from all 100,000 neurons at once, but maybe you can record from, say, 6,000 to 10,000 at a time in these horizontal slices. Okay. But that leads to a slight challenge, okay? So for every slice, we're actually getting kind of, for the samples we've recorded from, say these are the time series from the recording that we get, we actually are recording from different neurons. Now, there might be some overlap between these, between the neurons recorded in each layer, but there's clearly some pairs of neurons that were never recorded simultaneously ever, and that we can't, just because of physically where they're located in the brain, we can't record from altogether. So the question so basically here we have a chunk of the pairs of neurons that we have joint data on, and we're going to call this O, our observed set of neurons that we've recorded from jointly. And we're going to call everything in gray here O complements. So this is specifically where we've got no joint data at all. We basically have no observations, no measurements jointly of these neurons whatsoever. And the question we're going to ask, and we call this the graph quilting problem, is can we estimate a probabilistic graphical model from this partially observed data, where we don't observe all interactions between neurons, we only observe a subset of them. Another way of putting this, to make this kind of, even kind of bring this home at the fact that this is really challenging is if you look at this, we can't even estimate a marginal dependence. We can't estimate a correlation between pairs of neurons that we've never observed together at all, right? So if you can't even estimate a correlation or a marginal dependence, how on earth can you estimate a conditional dependence where we need to condition on all the observed neurons in order to estimate this? So, why do we call this quilting? Well, a couple of reasons. It sort of looks like a quilt, and there's different ways of different observation patterns that sort of look like plaid or quilt, but it's also related to covariance stitching, which is kind of completing a covariance. It's reminiscent of that. But so you might say, well, the graph quilting problem, can we estimate these probabilistic graphical models when we only have partial data? The answer, with no additional assumptions, the answer is an absolute no. And in fact, when we first started studying this problem, we spent a long time and trying to find a way to prove that it was possible to do, and we kept running into roadblocks, and we thought the answer was no for about 6 months before we discovered that you can do something. But this is a completely new problem. Nobody studied this before at all, and it seems impossible to do. It seems completely impossible to estimate conditional dependencies when you don't even have marginal dependencies. But we're going to actually show something rather cool. We're going to show that if this is the true edge structure from a graph, so here I'm basically plotting the P neurons by the P neurons. These dark points here are denoting an edge in the network structure between, say, neuron 1 and neuron J here, okay? So this is the true graph structure, and here I'm showing the kind of observation patterns that we have, the type of kind of observation sets we have, and we actually are going to show something kind of striking. For all pairs of neurons that we've observed jointly, we can actually perfectly, under certain conditions, perfectly estimate the graph structure if we've observed them jointly. If we haven't observed those pairs jointly, we can find a minimal, so first of all, the graph is not identifiable in these gray areas here in O complement, but we show that we find the minimal superset of edges that outline all possible graphs consistent with the O complement part. So we basically find the graph that is the best you can possibly do on this part. So this sounds crazy that you can estimate graph structures when you don't even have marginal dependence. So let's just really briefly, I'm not going to go into any of the math here. Oh my goodness, there's a lot of math. But I just want to kind of highlight the intuition for like how we do this and go about this. So first, just some brief setup. We're going to assume a Gaussian graphical model. So our first work on this is solely assuming Gaussian graphical models. We can probably extend this a little bit later. So there's a quick question in the chat. So recording neurons, so this is typically motivated by fluorescence imaging, not electrophysiology. But it could certainly be used in other settings as well. But we're typically motivated by 2-photon calcium imaging. So we're going to assume a Gaussian graphical model, and Gaussian graphical models are really nice to work with mathematically at first because the inverse covariance matrix or the precision matrix, if there are zeros in the off diagonal, these directly correspond to conditional independence statements. So these are really nice to work with. And we're just going to call our true edge set E, the observed pairs O, and sigma O kind of our observed true covariance matrix. So the first thing is, you might ask, when is it possible to exactly recover the graph from our observed covariances? So it turns out that only if our true edge set is contained in those that are observed, then we can recover the truth. So this basically means in our brain cube that, in our kind of slices that happen, that there can be no connections between neurons that were not recorded simultaneously. Well, we know that's dumb, and we know that's not true, right? So this result basically says that, oopsies, like, maybe there's not much you can do. You can only recover the graph when the true edges are only between pairs that you've observed. So this is clearly a problem. right? And, but we use this as motivation and propose a method that we call MAD-GQ, in part because it's MAD, but it also comes from the max determinate completion of a covariance that was introduced in the 70s, actually. And the idea of this is, what if we do maximum likelihood estimation, but we observe that all of the neurons we haven't recorded from simultaneously, we just assume that there's no edges in the graph. We assume there's no connections between the neurons that we haven't recorded from simultaneously. So this is clearly wrong, but let's just assume this and fit maximum likelihood anyway, right? And see what happens, okay? So when we do this, okay, so in this situation where the true edge set is actually in all of the observed nodes, and you fit the MAD-GQ, it does perfectly, of course, because these are the assumptions. So yay, we get perfect graph recovery here. But what happens when there are some edges that are not in the pairs of neurons that were jointly observed? So this is the MAD-GQ solution now. Suddenly we have so many false positives shown in green, and we've got the false negatives in our O complement part, the part that wasn't observed jointly here. Okay, so we clearly have a lot of false positives and a lot of false negatives there. But we actually get some of the graph structure in O correct. Why? What's going on? So it's actually really cool. We can prove that there are no false negatives with high probability in O using our MAD-GQ method. This is actually a really cool result. So we only get false positives, we don't get false negatives, meaning we don't miss any connections, okay? But can we go a step further here? And the answer is yes, we can actually do a lot better. So it turns out, and under some assumptions, and the assumptions are basically that our edges in our unobserved part connected to the observed part cannot be too strong, meaning the edges between our unobserved and our pairs of neurons that aren't observed and those that are observed can't be too strong. And the overall edges in the graph structure have to be bounded away from zero, meaning the edges have to have enough signal, they can't be weak. And under these conditions, we can take all of these false positives that are in green, and we're just gonna hard threshold them to zero. So it turns out we proved that all of the false positives that happen in green are really small in magnitude. So this is actually really cool. So all of the green stuff under these conditions are very small in magnitude, and you can easily pick a threshold, and the threshold, it's fairly robust. We have explicit bounds in the paper for different graph structures, but you can very easily find a threshold that will give you, threshold all those false positives away and give you perfect graph recovery for all pairs of neurons that were jointly observed. So this is really cool. Suddenly we've got a perfect graph in the observed part. This sounds crazy. This sounds even hard to do, but we have this. So this is really cool. But you're gonna ask, okay, well, what do we do about all the false positives in the unobserved pairs of neurons? Like, if we don't jointly observe pairs of neurons, is there a way to estimate a graph structure? And again, the answer is yes, we can actually do something even though we don't even have a correlation between these. So what we're going to do is we're going to actually use some of the information in the observed part to do this. And I'm going to just illustrate this with a quick picture. This is a simple graph structure here. I'm showing you the MAD-GQ solution where, again, false positives are in green and false negatives are in red. And what happens here is that every time we have a false positive for a particular neuron on the off diagonal of that particular neuron, we know that that false positive can only occur if there is an edge in O complement somewhere between pairs of neurons we haven't observed. From conditional dependence relationships and other assumptions, this is the only way in which we get an edge there. And so what happens is all of these green dots show us where all of these greens right here show us exactly where there are potential edges in O complement, and we can take the intersection of these, and suddenly we get these. Okay, so the graph is not identifiable between this edge and this edge, or this edge and this edge right here, meaning that there's no way from the data that you're given to identify which edge is it, kind of this pattern, this diagonal pattern, or this diagonal pattern of edges. But we can get this minimal superset, and we show that this superset is minimal and that you can't reduce this further and still have a graph consistent with the observed pairs of neurons. Okay? So this is a minimal superset, and at least we've eliminated lots of edges and O complements from possibilities. And we can certainly use this. Oh, and further, we actually know we have some results about the number of edges and O complements, bounds on the number of edges. So you can still use this to study the functional connectivity structure, even if you don't have all of the information. You only have the part that's identifiable in O complement. So further, this was a simple example. You can go a step further and identify where each of these edges are, this minimal superset, using a recursive complement algorithm. So this is the RICO algorithm. I'm not going to go into any details at all, but using this, we can identify this minimal superset in O complements of edges. Okay. So, and this algorithm, I've shown you this for this kind of block structure that you would get if you record from horizontal slices one at a time in the brain cube. But this procedure actually works for any type of observation pattern. You don't just have to have changed blocks. And you can see here from applying our method, the grays are all the unobserved portions, and you can see that we have no false negatives, but we do have some false positives in the unobserved portion. So this is actually still a really cool result, because what this allows you to do is this allows you to recover largely the graph structure with no false negatives. You will have some false positives, but we know about how many overall edges there are. So we can still answer some of the questions about what are those functional connectivity patterns in the brain, how is functional connectivity related to structural connectivity, even if we don't have joint observations for all of those pairs of neurons, okay? And I should say, everything I've shown you is kind of the population case. So this is assuming that all our model assumptions are true, which of course they're not, and that we have infinite data. Now, of course, we don't have infinite data, and you can estimate this in finite samples. I'm going to kind of skip this for now, but we get rates similar to the rates that you would get for the high-dimensional analysis of the graphical lasso. And this is basically taking our population analysis and our results there and using kind of those high-dimensional graphical lasso results to get these types of bounds. So these tend to hold under certain assumptions with high probability in practice. Okay? Cool. Now I want to show you a quick kind of application to functional connectivity. This is from a Stringer et al. Data set on, this is GCamp 2-photon calcium imaging. There's over 9,000 neurons in this study. This is a publicly available data set, so other people can go and kind of look at this. These are kind of a quick visualization of the cube from this data, and it was recorded at kind of these horizontal slices in the brain, just like the motivation in the setup here. Okay, so I'm skipping. So the part of the experiment, there was some kind of there was some stimuli, but we actually were focused on spontaneous activity portion. So there is quite a bit of recording of spontaneous activity, and we're using this for this functional connectivity estimation right here. So the first thing we did is we're going to use this data to have kind of a realistic inspired simulation study so we can assess how well our method is doing. And so the first thing is to take all of these neurons and fit a graphical model to estimate kind of the functional connectivity between all of these neurons. So that's step number one. We have a graph estimate for the real data. Now, from this graph estimate, we simulate multivariate normal data from a Gaussian graphical model, and so that we know the truth, basically, and can assess how well we're doing. And we look at the percentage kind of pairs of neurons that weren't observed, and we do two situations, kind of 20% and 40% of the neurons that aren't observed. There's a question there, is there any way to include directionality? So in Gaussian graphical models, these are undirected graphical models. There are ways to estimate directed graphical models, and that's possibly an extension of our work. We have not looked at that yet, but so far we're really focusing on undirected connectivity. And most people do this in calcium imaging. That's the more common type of connectivity to look at. So again, in this simulation, we have we simulate kind of this pattern, and we've got some either 20% or 40% of the neurons missing. And what we're doing here is, as a function of the sample size, or basically the number of time points that we're recording from, we're reporting the AUC, area under the curve. So again, 1 is perfect recovery of the graph, and so forth. This blue line is when there's no missingness, meaning there's all of the pairs of neurons have been completely jointly observed. You're not doing these slices of the brain cube. And this is how well the graphical assay does without any missingness. These dashed lines and dotted lines show if we've got 20% of the pairs of neurons unobserved, or 40%, how well we do. And notice here, we actually don't lose that much accuracy. We're actually pretty darn close to as if we had all of the data. We are not losing that much accuracy. And again, this drop comes typically only from false positives, not from false negatives. So we can do pretty darn well and get a fairly accurate estimate of the graph, even if we don't have the full data kind of jointly observed. And here I'm just showing you another example. So this is one horizontal, so we can zoom in because it's a little bit hard to see the whole brain cube. This is a zoomed in version of the estimated graph from one horizontal slice only of the same data set. And this is if you fit just the regular probabilistic graphical model, this is an estimate if we observed all of the data. And here is if you're actually missing some of the joint observations. The gray portions are 20% missing or 40% missing of these observations here. And in blue, we're showing you false positives, and in red, false negatives. And when there is 20% missing or 40% missing, there are some false positives and false negatives. But I just wanted to show you this visualization to show you that we actually largely get the structure of the network and the properties of the network pretty darn close, actually, even if we've got all of these pairs of neurons that are missing. And so just to kind of summarize, graph quilting is a completely unstudied problem before. It's motivated by problems in neuroscience. Now, the first paper we wrote is largely a theoretical paper, but we do have demonstrations on calcium imaging data. But there's so many open problems related to this. And it's kind of cool that you can estimate a graph and have provable guarantees that you can estimate a graph correctly, even when you can't even estimate marginal dependence or correlation. So it's kind of a fun problem and a cool result. And certainly we showed applications to neuroscience, but there's actually, when we started thinking about it, there's lots of other potential applications, including in genomics and other fields, where you don't always have observed all of the data at once. And because of that, you actually have this graph quilting problem comes up quite a bit. So I'm going to briefly conclude here. We have acknowledgements. First, my collaborators, Giuseppe Vinci and Gautam Dasarthi, who were great on this. This was funded by NSF and NIH, and we do have a preprint available on arXiv, and it's currently undergoing some revisions, and hopefully we will be updating that shortly. And thanks very much for your attention. And I'm happy to take any questions anybody has.
Thanks so much, Genevera. That was great. I really appreciated it. Looks like we do have a question in the Q&A from Clay. So he points out that GCamp has a several hundred millisecond time constant. And so, how important is quilting if you're sort of sampling maybe on the order of that time constant between the different blocks?
Yeah, so that's a really good question, and we haven't quite worked with that yet. So right now, the data we're working with was kind of corrected for any of those time delays between the horizontal slices. And I believe the stringer data was collected at different horizontal slices. The data from the Tolias lab that we're working with was interwoven. So like sample 1 might have 4 vertical slices like this, and sample 2 is like this, and it keeps moving down like this. So I think that is definitely room for future work to consider stuff like that. We have not done that yet. Other questions?
Yeah, I'll follow up on. Please, folks, type your questions in either to the chat or the Q&A. It'd be great to. We have another 12 minutes or so. It'd be great to use the time. I'll have a quick follow up on that. So you pointed out that in the stringer data, you looked at the spontaneous activity, and I can imagine that things become really hairy when you have non-stationarity in the statistics. Especially between blocks. And I've wondered if you've even started to think about the way to handle that problem. And obviously that comes up in the context of stimulus, but in other contexts as well.
So I certainly think that would be really, really hard to do if every one of your recording blocks has like a different experiment, right? That would be nigh impossible, I would argue, right? So, I mean, and maybe you can do this, and one way to do this is to make sure you have just some kind of free time at the beginning of spontaneous activity before you start your experiments. But the other thing is we are kind of working with some data that we do have stimulus, but it's the exact same experiments run in different sessions, right? So there's all these different sessions, and it's the exact same experiment run. And I think there's a lot of different ways to account for the fact that you've got stimuli. Right now, we're just estimating kind of latent effects. We're kind of trying to regress out the effects of the stimuli and regress out kind of latent effects of the brain that are kind of in response to the stimuli, and essentially trying to fit graphical models to the residuals of after we've kind of adjusted for all of the stimulus related activity. And one of the good things when you do that and you kind of adjust for all the stimuli is that your data looks, hopefully, if you've done a proper job, slightly more Gaussian, and Gaussian graphical models are more appropriate then. So that is kind of an added benefit. You can also adjust for, in calcium imaging, of course, there's like autoregressive effects, right, in the signal, and you can, of course, adjust for those as well, and kind of first, and then all of the residuals you get left over, you say that actually they're correlated residuals, and the correlation is coming from the functional connectivity between the neurons.
Makes sense. Well, hopefully we'll get another question or two from the audience, but I'll ask another one since we haven't heard yet. I was thinking of, I have no idea about what's going on under the hood with your approach, but I was wondering if you could maybe try to give us a little intuition as to why the false positives are small and can be thresholded away.
Yeah, so it turns out, I'm gonna try to go back to some point at which, okay, false positives here. Yeah, so it turns out that the false positives, you can show false positives are only produced when there is kind of, you've missed an edge basically in the pairs of neurons, O complement, the pairs of neurons that you haven't observed. So what this really means is that the false positive is produced by like all of the paths going from this neuron to this neuron that go through this neuron here. So because of that, they're actually, if these weights are relatively small, then all of the paths coming in, actually the difference here is relatively small from 0. And because of that, you can show that these are actually typically quite small. So for these accompanying figures that we have in the paper and some more simulations, we've actually shown what the bounds are basically as a function of the maximum edge value in O complement, the pairs of neurons that we haven't observed, and the minimum edge value in O. You can actually show kind of how large those values are. And in most cases in a graph like this, there's a very simple value of tau for which you can do this. So it actually does seem, these assumptions seem to hold in practice, at least in simulated networks that we've looked at. It seems to hold fairly regularly.
Does it look bimodal then in the data, in the neural data, that you see a set that's substantially smaller than the remainder?
Well, I mean, with real data, it's always hard because you kind of, I mean, you never know how many true edges there are, what the true edge weights are. And largely this result, the ability to threshold the false positive away only happens with very sparse networks that are relatively strong in signal. So if you can think about, even if that doesn't necessarily hold for neural data, but you can think about the strongest functional connections, hopefully are represented as a sparse network. Maybe it would hold for those, which is what you care about anyway, right? I think that's the hope.
Sure.
Right?
Looks like we got a couple more questions. I'm not sure I completely understand the first. How does the inference time scale with the data size? The inference time scale with the data? I'm not sure what exactly, maybe.
Inference time, I guess I'm a little, do you mean the computational time? The computational complexity to run this algorithm?
So maybe LR, if you could just clarify your question, that would be great. In the meantime, let's look at this one from Sankar. Is it possible to use this approach in oscillatory signals to get frequency-specific graph models?
Ooh, I mean, that's really interesting and really hard, but I do think it's doable. I happen to know someone, a former postdoc at Rice who, no, he's a former PhD student at Rice now, he's a postdoc elsewhere, who is working on complex graphical models in the frequency domain. And I do actually think that you can use this with those. That's a very, very fun and hard future paper to write. But then, yes, I do think you could use that in ECOG type stuff, and that would be really interesting in ECOG. Yeah.
Cool. Okay, so yeah, the question from LR was about algorithmic complexity.
Algorithmic complexity. Oh, yeah. So is this algorithm fast? The answer is, to be honest, not really. But when you've spent that much money to image 100,000 neurons anyway,
what's
like,
another GPU would do at that point?
Does it matter that much that we're taking like several hours to several days to run when you've already spent millions and millions of dollars gathering the data? I would say no, and it's okay. This is not going to be a fast algorithm, right? But it does come with really good theoretical guarantees when we can get this to run.
And we got one in the chat. Does an edge imply dependence at 0 time lag or in general dependence?
Yeah, so these edges are all basically 0 time lag or kind of contemporaneous connectivity, right? So not lagged connectivity, not directional connectivity, contemporaneous connectivity. Which in calcium imaging, because it's at a slower time scale and you can't resolve individual spikes anyway, I would argue is probably the best you're going to do as far as functional connectivity. I think there are potential other ways to extend these type of results if you do want to look at lag dependence or kind of Granger causal dependence and directed graphical models. But that is not something we've looked at yet.
maybe I'll throw another question in. I'm sure you get this with some regularity. What does functional connectivity tell us about synaptic connectivity?
So I don't know the answer to that yet. I am hoping, in part because it is very hard to get data on synaptic connectivity and functional. There is some, and I'm hoping to work with it and hopefully be able to answer that question in the near future. But I do think that's an open question, right? And I mean, our hope is like maybe we can't get the exact edge structure of kind of the structural connections, but maybe we can get some of the same properties. And if so, we can still use functional connectivity to get inference about properties of kind of the structural connectome, which would still be scientifically interesting.
Sure. I guess the idea being, can you use the graph you infer to understand things about computation?
Yeah, yeah.
Okay, well, last chance, folks. Anybody else got a question for Genevera? Otherwise, thank you so much for your time and for your presentation. And thanks to everyone in the audience for joining and participating in Neuromatch this week. There's a lot more content, tons, if you go on the website of talks that are going on all week. And hope to see you at more events soon. So thanks a lot.
Thank you very much for having me. It was great to see you, and I really enjoyed it.
Yep, take care.
Thank you. Bye.